Okay, so in this video, I'm going to talk about aerobic cellular respiration. Remember, aerobic cellular respiration happens in the mitochondria of eukaryotic cells. It happens in the presence of oxygen, which is why we call it aerobic, and it's going to produce the majority of ATP in our cells. Um, that allows us to kind of do cellular work. So um, the first thing that's going to happen in aerobic cellular respiration or in any version of respiration, both anaerobic and aerobic, is glycolysis. So this first step of aerobic and anaerobic respiration is glycolysis. Remember, if there's no oxygen present, then after glycolysis, eukaryotic cells can do fermentation. um, if the fermentation occurs in muscle cells of humans, it would be lactic acid fermentation. This results in the buildup of lactic acid, which causes your muscle cells to start to burn. So this is why when you're exercising, if you're feeling the burn, you are feeling the buildup of lactic acid, which is telling you to slow down and take a breather so that you can accumulate oxygen and you can switch back to aerobic respiration, which is more efficient and gets much more ATP per unit of glucose. Um, Also, bacteria and yeast can do anaerobic respiration. They are able to do that through the process of also alcoholic fermentation, which is going to result in the buildup of ethanol. So, glycolysis is going to occur in the cytoplasm, so it can occur in both eukaryotic organisms and prokaryotic organisms. But in this video, we're going to focus on aerobic respiration that occurs in eukaryotic cells in the mitochondria. So, after glycolysis, then this process will enter the mitochondria and we will break down glucose fully and make the majority of the ATP for our cell. So, during glycolysis, our 6-carbon glucose is going to be broken down into two 3-carbon sugars called pyruvate. So, the first thing I'm going to do is track the carbon as it moves through the process of respiration and kind of see where the carbon goes, and then we're going to track the flow of electrons. So, the whole purpose of cellular respiration is to break down glucose. And as we're breaking it down, we are breaking bonds. So if you remember from chemistry, breaking bonds involves breaking bonds that are made out of electrons. So glucose is made of a covalently bonded molecule, and in a covalent bond, the electrons are shared. So every time you break a bond, you're going to release the electrons that were stored within that bond, and those electrons, I like to think of it as them kind of like spilling all over the place, and they need to be picked up. So they're going to be picked up by electron carriers. Well, electron carriers, remember, those are like, I call them like little Ubers, and the purpose of them is to shuttle electrons to the electron transport chain. They're going to pick up the electrons that got spilled all over the place, and they're going to transport them to the electron transport chain. The electron transport chain is going to be the money maker, or where the majority of the ATP is made. So every time we break a bond, electrons are released, picked up by electron carriers, and brought to the electron transport chain. Once they arrive at the electron transport chain, they can be used to create a proton gradient or a proton motive force that can then be harnessed through ATP synthase to create ATP. So, in this right here, when we start with glucose and we break it down into two 3-carbon molecules, we started with 6 carbons. We've broken it down into two 3-carbon pyruvates. That other pyruvate's not shown, but it's there. We have not lost any of our carbons yet. So we have broken a molecule from a 6-carbon molecule to a 3-carbon molecule, but we haven't lost any carbons. So again, the first thing I want to do is track the carbons. So let's do that. So here we have a 6-carbon glucose, which is going to be broken down into two 3-carbon pyruvates. So we still have 6 carbons total. This is going to diffuse across the mitochondrial membrane into the matrix of the mitochondria. And before it can enter the citric acid cycle, it needs to be broken down into a 2-carbon sugar called acetyl-CoA. So here we're going to go from a 3-carbon molecule to a 2-carbon molecule. Well, when we did that, we did lose a carbon, and the way carbon is released from the cell is through a gas called carbon dioxide, which is, of course, what we exhale when we breathe out. So this 2-carbon sugar is going to be further broken down through the citric acid cycle, sometimes called the Krebs cycle. And as we break it down, we're going to release the remainder of the carbon that was stored in the original pyruvate in the form of carbon dioxide. So here we had a 3-carbon sugar. It was broken down or oxidized into a 2-carbon sugar, and that is going to release 1 carbon dioxide. And then as the remainder of the 2-carbon molecule is broken down in the Krebs cycle, we release 2 more carbon dioxides into the atmosphere. So now all of the carbon that was originally tied up in the original pyruvate is going to be released as carbon dioxide into the atmosphere. Remember, this happens 2 times because we made 2 pyruvates when we broke down the 6-carbon sugar. So the whole purpose of cellular respiration is not to create carbon dioxide, because as you can see, carbon dioxide is just removed as a waste product. It's going to build up inside the mitochondria. It's going to diffuse out of the mitochondria, eventually into the cell, and then it'll build up in the cell and it'll be released into the blood, where it can travel to the lungs and be exhaled by our body. So we don't really even want to create carbon dioxide. We want to break down the glucose, because what's in the bonds of glucose? Electrons, and the electrons can help us create kind of this proton motive force that allows us to create ATP. So electrons are very important, and that is the whole purpose of breaking down this glucose molecule. It's important that we create carbon dioxide and that we exhale it into the atmosphere, because that carbon dioxide can be used in photosynthesis. But we do not need that in the process of cellular respiration. It's exhaled as a waste product. Okay, so now that we've tracked our carbons, we've released all the carbons that were originally in glucose. Let's track our electrons. So as glucose is converted into pyruvate, we didn't lose any carbons, but we still broke a 6-carbon molecule into 2 3-carbon molecules. So we broke a bond, and so, or several bonds. And so to do that, we had to release electrons, which were going to be picked up by electron carriers. So here is the electron carrier containing an electron, and it is going to travel to the electron transport chain. Then pyruvate is going to, again, diffuse through the mitochondrial membrane. As we break down this 3-carbon pyruvate into the 2-carbon acetyl-CoA, again, we go from a 3 to a 2-carbon molecule, so we've broken another bond. And so to do that, we had to release electrons, which are picked up by an electron carrier and brought to the electron transport chain. As we completely oxidize glucose, or completely break down the remainder of glucose that is stored within the acetyl-CoA, we're going to release the remainder of the carbon, but we're also going to release the remainder of the energy stored in those bonds in the form of electrons. So those will be picked up by electron carriers and brought to the electron transport chain. So the purpose of every reaction, of every oxidation reaction here, is just to extract the electrons and bring them to the electron transport chain. The purpose of the electron transport chain is to release energy. So the electron transport chain is a series of embedded proteins in the membrane, and the electrons are going to get passed through these proteins, and as they do, they are going to release energy. So this whole process is an exergonic reaction. And an exergonic reaction means, remember, that it releases energy. So this exergonic reaction is going to be the driving force needed to pump protons against their gradient. So these protons are going to be pumped against their concentration gradient from an area of low hydrogen to an area of high hydrogen concentration. And so this is going to create something called a proton motive force, or this stored potential energy of hydrogen ions. This pumping of protons against their gradient is an endergonic reaction. Sorry, that's kind of messy. Endergonic, kind of looks like enderganic. Okay, this is an endergonic reaction. So this is a form of energy coupling where we use an exergonic reaction to provide energy for an endergonic reaction. So we're using the release of energy from the electrons flowing through these proteins to drive the endergonic pumping of hydrogens across the membrane, again, from an area of low concentration to an area of high concentration. This is active transport. They do not want to do this. So here, as we create this source of stored potential energy, the hydrogen ions can then flow through the enzyme ATP synthase, and this is an exergonic reaction, because now they're going along their concentration gradient through the protein ATP synthase. And as they flow through their ATP synthase along their concentration gradient, we use that to phosphorylate ADP. And when we add a phosphorus or a phosphate to ADP to create ATP, that is an endergonic reaction. So again, this is a form of energy coupling where we are using the energy released from the exergonic reaction of hydrogen ions going along their concentration gradient. Then we use that energy that is released to fuel the endergonic reaction of the phosphorylation of ADP. Again, phosphorylation just means you're adding a phosphate group to this ADP to create adenosine triphosphate. So adenosine diphosphate plus a phosphate gives us adenosine triphosphate. And this is going to give us the majority of our ATP, which is probably about 25 to 30, depending on how efficient the cell is. So this is the majority of the ATP that we get in aerobic respiration. There's also ATP created in glycolysis. There are 2 ATP formed there through substrate level phosphorylation, and there are 2 ATP formed here. Now, this process is called oxidative phosphorylation, and the reason it's called oxidative phosphorylation is because it is driven by oxygen. Now, why are these electrons flowing through the electron transport chain? What is the driving force of the electron transport chain? Well, the driving force is oxygen. If you remember when we talked about water, oxygen is highly electronegative, meaning that it strongly attracts electrons. This is why water is polar, because oxygen attracts electrons more strongly than hydrogen does, creating a partial negative charge on oxygen and a partial positive charge on hydrogen. So oxygen is still highly electronegative, and as it pulls electrons through the electron transport chain, they release energy, which again provides the necessary energy to pump protons against their gradient. So oxygen is the driving force of the electron transport chain. If you do not have oxygen, the electrons will not be pulled through the electron transport chain, and they will not pump protons against their gradient. And without a proton gradient, you're not going to be able to create the majority of ATP that is created during aerobic respiration. So it's really important that we have oxygen in order for this process to occur, which is why when we don't have oxygen, when we're exercising a lot, we are going to switch to anaerobic respiration, and we're going to take glucose and we're going to break it down into pyruvate, and then from there, we're going to convert that to lactic acid, and that's going to cause the lactic acid buildup in your cells. That only creates 2 ATP. So your cells still have a little bit of ATP to work with, but it's not nearly as much as if there were oxygen present. That's why the lactic acid causes your cells to burn, tells you to slow down so that you can regain your oxygen, so that you can convert back to aerobic respiration. Now, all the electrons that are going to then join up with oxygen are going to pick up a couple hydrogen ions that are floating around, and we're going to create a water molecule, which can then be used by the cell or excreted by the cell, depending on where it's needed. So all the electrons that went through the electron transport chain eventually end up with hydrogen. So the purpose of cellular respiration is to break down, or we could also say oxidize glucose. And the reason we're oxidizing glucose is to obtain the energy that is stored within the bonds of that carbohydrate. It's really high energy carbohydrate. We're going to release the electrons that are stored in the bonds. Those electrons are going to be picked up by electron carriers. The electron carriers are going to travel to the electron transport chain, and they're going to travel because they are attracted to this highly electronegative atom of oxygen. And as oxygen attracts the electrons, they're going to release energy in an exergonic reaction, which is used to power the endergonic pumping of hydrogen ions across the membrane. And then the pumping of hydrogen ions creates this proton motive force, which can then be harnessed through ATP synthase as they go along their gradient, is an exergonic reaction, which can then allow the phosphorylation of ADP to create adenosine triphosphate in large quantities of about 25 to 30 per molecule of glucose. So that is aerobic respiration. Other things I've seen them like to mention or kind of throw at you is what would happen if this membrane was punctured or leaking. Well, remember that if the membrane is not able to help create this gradient, then the hydrogen ions would just flow back down along their gradient, and we wouldn't create this proton motive force, having them harnessed where they do not want to be. And if we couldn't create this potential energy, then they couldn't, it would not be an exergonic reaction as they go through the membrane. It would not release enough energy to phosphorylate ADP, because they would just be leaking back all the time. You wouldn't create a gradient. They would probably just be like an equilibrium and everything would be fine, but nothing could be harnessed for energy. And then you wouldn't be able to use this proton motive force to create ATP. So we're using all these energy coupling reactions in order to make cellular energy. And all of these are ways that we're able to make ATP without using cellular energy in the form of ATP, because it wouldn't make any sense to use ATP to make ATP. So we have to find other sources of cellular energy. And in this case, we're coupling exergonic reactions with endergonic reactions in order to create the ultimate energy molecule that most cells rely on, which is ATP. So I hope that helps, and I hope that makes sense. And good luck studying.